Now, we got into this area by virtue of the fact that we became the first group that developed antibodies to the extracellular domain of PSMA, and we first published that a little over 10 years ago. And one of those antibodies, which we call J591, we genetically engineered to be a humanized antibody so that we could use it in clinical trials. Because we had the first antibody that bound to the extracellular domain and we could use it to study viable tumor cells in vitro, we also found unexpectedly that PSMA was internalized, as I've already mentioned. I say unexpectedly because when it was originally sequenced, it did not demonstrate any known internalization motif. We subsequently defined a novel internalization motif. So our clinical strategy, again, simply stated, is in one set of circumstances, we're targeting a radioisotope, taking advantage of the fact that prostate cancer is a radiosensitive tumor. We have a whole other series of studies where we're targeting highly potent drugs, but because of the time constraints, I'm just going to talk about the radioisotope work. We can use radioisotopes because prostate cancer, as I said, is a relatively radiosensitive tumor. We have the additional benefit that the drug of standard therapy for patients with metastatic prostate cancer is called docetaxel, and this has a radiosensitizing function, so there's an obvious potential synergy that we can obtain by, at some point, combining our radiolabeled antibodies with docetaxel. But more to the point, the area that we're really interested in is in targeting a very large population of prostate cancer patients who have failed primary management of their localized disease. They've had surgery or they've had radiation therapy, and roughly 30% of those patients will ultimately show signs of failure, initially evidenced by rising PSA, and those failures are due to the fact that at the time of initial diagnosis, those patients harbor micrometastatic disease, generally in the bone marrow. And this is a population of at least 750,000 men in the United States. So it is a large patient population, and it is an ideal setting for potentially curative therapy. When we began these studies, we had the option of using either of two radioisotopes, yttrium-90 and lutetium-177. Yttrium-90 had already been approved as a targeting isotope on a different antibody for the treatment of non-Hodgkin's lymphoma, and in that case had exceedingly terrific clinical benefit with very high response rates. I don't want to spend time going into the different physical characteristics of these two isotopes, but I do want to point out one thing about the targeted delivery of isotopes, which is different from any other type of systemic therapy, and that is that isotopes have their physical characteristics are such that there is a range of tumor sizes that a given isotope is ideal for. And in the case of yttrium-90, that's really much better suited to larger volume disease in the range of, as you see on the slide, 28 to 42 millimeters, whereas lutetium-177 is much better for disease in the range of 2 millimeters, which again is earlier than one would typically see on a conventional CAT scan, MRI, or bone scan. This was initially determined by Joe O'Donoghue at Sloan Kettering, who demonstrated, as you can see in this figure here, that if a tumor lesion is too small for a given radioisotope, the energy of that isotope gets delivered outside the tumor, as opposed to in the tumor. And conversely, if the tumor is too large for a particular isotope, you don't get adequate penetration. There are various ways to solve this problem, and Joe O'Donoghue pointed out the fact that you can actually combine different isotopes and get much broader cell kill, but that's again beyond the scope of what I'm going to talk about today. I just want to briefly point out the fact that we've done a number of clinical trials using this particular antibody. We're on our 11th clinical trial now. We've treated over 300 patients, so we have a significant body of clinical data. If I focus just on the radiolabeled antibody trials in prostate cancer, we're approaching 150 patients now. We've completed the phase 1 studies of each of these isotopes attached to the J591 antibody. Both of those studies have been published. I'm just going to summarize those briefly by saying that by this approach, the treatment is very well tolerated. Patients have essentially no symptomatic side effects whatsoever. We saw virtually flawless targeting of tumor sites in virtually every patient, and I'll demonstrate that for you. The genetically engineered humanized antibody has been non-immunogenic. We can treat patients multiple times. The dose-limiting toxicity with radioisotopes in general, and it's no different with this radiolabeled antibody, is a decrease in platelet count. We've determined the maximum tolerated dose of either of these isotopes in combination with the antibody, and even in the phase 1 trials we saw significant evidence of biological activity. So moving on to the phase 2 trial, we felt that we could not continue using both these isotopes in parallel. It wasn't practically possible. So we decided that we would select lutetium-177 because this is the isotope with the shorter range, and this is the isotope that would allow us to treat those 750,000 men in the United States who have rising PSAs prior to the onset of frank metastatic disease, and these are the people that we felt we had the greatest opportunity to cure with this approach. But having done just one phase 1 study in a limited number of patients, we didn't feel that we were in a position to go immediately to those patients for ethical reasons, and so we had to stick with, again, as is typical in these early phase clinical trials, we had to stick with patients in their late stage of disease who had no other treatment options available to them. And even though these were not ideal candidates for this particular isotope, we needed to get more toxicity data. And so in the phase 2 trial that I'm going to describe to you, all of these patients had progressing, hormone-refractory, frankly metastatic prostate cancer. These patients were allowed to have had prior chemotherapy. We imposed no limitation on the extent or bulk of their disease. In none of our trials have we attempted to select patients on the basis of PSMA expression because of what I've already told you, which is the dogma is that every prostate cancer is PSMA positive. And all of the patients in this trial got a single dose of lutetium-177, J591. There were 32 patients in this trial. Because this was the first phase 2 trial in the United States that used lutetium-labeled antibody, even though we had previously determined that the maximum tolerated dose was 70 millicuries per meter squared, the FDA required us to do a cohort of patients at a somewhat lower dose to confirm that, which we did.